And it's our priority to know something about these animals, why they have been doing such kinds of things, and where does this comes from. And that word we have given it as zoonotic. Zoonotic means the disease that transfers from animals to human beings. We call it as zoonotics. And I am going to share my ideas on the bats, mainly on two structures. One is on the ethology, and that is the behavioral biology of bats, that what we are doing, and also on its importance. We have been doing on conservation strategies for the past 20 years, mainly on how they are useful to the human beings and how we are benefited by bats. So with this slight introduction, I am moving on to this one. If you take down this one, there is category, there is vertebrate animals, invertebrates, plants, that is the living things, what we are well known and aware of in this biology webinar. I thought most of you will be biology, depending on biology subjects. If you take down, there are around 5,500 species of mammals, around 10,000 species of birds, and 10,000 reptiles, and of course the invertebrates that are sumptuous amounts, the arachnids, insects, mollusks, that has been spread throughout the environment at a large rate. And also the plants, we have not included the algae and other small plants, but here we have given it as flowering and also the conifers that has been spread throughout the environment. In the right category, the world is spinning. If you see there will be a small red line that we have given, and what it denotes is the equatorial line. You may be well aware of that one. What is the importance of this world? If you look after this globe, we can share many ideas just by looking after this one. When we look after on the left side, we have given so much of things, that is the vertebrates, invertebrates, plants, and animals. But where are they and how they have been important to us, and how we are getting the most benefit from these animals, and where they are? If you take down the equatorial line, we call it as the tropical area. In the globe, the tropical area where it has been present is the equatorial line. And in the subtropics, that is the adjacent to the equatorial line, we call it as the subtropics. India is a subtropic country, and the diversity of animals will be very much and in large amount in this tropics and subtropics. If you take down, if you go towards the polar region, the diversity of animals will get lesser and lesser and lesser. The altitude differs, and the variation also differs. If you take down, if we come out of our house and look after the plants, they are green in color, of course. But how many greens are there? There will be more numbers. The diversity will be more. The genetic diversity will be more, which we can't find in any of the other temperate countries. That is behind that tropical areas, there will be temperate countries. There the diversity will be very, very lesser. Even we can take it in any of the places. If you take down in the Western Ghats, where we are doing a lot of research, in the lower ranges or the base of the Western Ghats, foothills of Western Ghats, there will be more number of species. If we climb towards the Western Ghats, the diversity and the species of animals will get lower and lower. So the diversity will be more. There has been more things to be studied on this equatorial line, because we know the rainy season is going to come. The air has to come from the polar regions to this equatorial line. That is, it gives the globe gives the complete structure of the nature, how we have been benefited, how these animals are spread, how they are diverse. Such a phenomenon is there behind this equatorial line, and how many water currents are there in this world. So the water currents has to do their work, then only we can get a lot of rain, and all other things has to be spread on at various times. With this introduction, the bats, of course, the mammals, we are well aware of it, they are mammals. And they have been classified into order Chiroptera. In the old word classification, we have classified into Megachiroptera and Microchiroptera. Megachiroptera, we denote it, they depends on plants and animals. If the species of bat that depends on animals, we call it as Megachiropterans. And if it depends on other than plants materials like insects, frog, bloods, or any other animal matters, we call it as Microchiroptera. They make up around 20% of the total numbers of bats, and they are the second largest next to rodents. Rodents are the first category in mammalian diversity. The next to the rodents, the large number of species diversity was present in bats. In India, there are about 123 species of micro and mega chiropterans. And we should know their diversity and also the habitat, how they have been, why they have been, and where they have been. At the start of my research in the 90s or end of the 90s and 2000, where I start my research, everyone asked me, why you are doing research on bats? There is so much of area. Why you have chosen bats? Actually, this is an animal that has to be studied, and it's a negligible species. Everyone don't like bats because we look after it as an ill omen. We don't know the importance and how they have been responding for us and giving benefits to us in various things. And I'm going to share my ideas on this one. Bats statistics, there is around 500 species of mammals, and of course there are 1,300 species are of bats. Of this, 88% are of tropical. They are long-lived animals. That is the important thing. I will come to that and share my ideas. The simple word long-lived gives various things that what we should know about bats, because they live up to 60 years. You can't all mammal to see, these animals live for around 60 years. How can a small animal can live for such a long years? The answer for all this question is, are bats zoonotics. They are having low fecundity because they lay 1 or 2 young ones at a time. They take care of their young ones. Of course, they are mammals. They have high survivorship. They are warm-blooded animals. They nurse their babies with milk. It has fur around its body, and it has been spread throughout the world. That's an important thing. Except Antarctica, it has been spread throughout the world. And how it has been spread throughout the world. That's also a thing has to be. If a species has to be successfully live in this environment, it should make itself adapted to the environmental conditions, and also with the food. So 2 things are necessary for an animal to adapt to an environment. One is how it has to be used the environment for its own. That's only bat has been spread throughout the environment. Certain animals are specific to certain places. Certain plants are specific to certain places. You can't see kangaroo except Australia in anywhere of the world. And how these bats has been present throughout the world. That is another question that has to be answered, and why they have been doing such a thing. And about bats, a simple phenomenon. They are fruit. If they feed on fruits, we call it as frugivory. They eat on flowers or plant leaves, we call it as folivory. And carnivorous, of course, they eat on birds, reptiles, and amphibians. Fish, they are feed on high specialist carnivores. They feed on fish. There are species that feed on blood of an animal or humans. We call it as sanguivore. And insects, of course, they are aerial and foliage gleaners, insectivorous species. Actually, when a bat feed on fruits or leaves, they won't totally chew the complete think of food. They just chew the fruits and take in only the juice of the fruits. They don't eat the complete thing. They spit out the remnants. That only everyone will use to say bat eat on with its mouth and defecate with its mouth. No, not like that. It has all the structures what a human has. It has been compared with the human. The structures are similar to a mammal. All the structures are there. Usually, what it will do means it will chew the fruits, drink the juice, and spit out the remnants. So it won't take the complete fruit as other mammals do. So the spill down part is called as the bolus. So there comes another thing. Keep in your mind, I will come to this word, small word bolus there, and we will discuss about this bolus in the later slides. And where bat lives, we call it as roosts. The roost has been classified into 2 types. One is diurnal roost, where it spends its day, and night roost, where it spends its night. We of course know bats are nocturnal in habit. They fly only during the night time, not during the diurnal time at anywhere in the world, because they have been dependent on nocturnal activities. And of course, when we think of these bats, why they are important, they are planters of seeds, they are pollinators, they disperse seeds, They are enemies to insects and control insect population at a larger rate. And it has been there and helpful for us in various things. They are enemies to insects because in various countries they have been used as a insect control agent at a larger rate. We can share it. So these are all of the few microchiropterids. The photos has been taken by us, which is around the Western Ghats. That's the plains of the Western Ghats. There is the Hippocidus furis, and in Tamil Nadu, of course, we are doing research in Tamil Nadu, and there is around 20 species of insectivorous species. And this one, there is the Hippocidus species. There is Megaderma lyra. There is a false vampire bat. There is a third one in the top. That is the false vampire. Here we don't have vampire bats that feed on completely on blood. It is present in the American countries. It is called as Desmodus rotundus, and it is a species that will feed completely on blood of small mammals. They feed on bovines, that is the cow varieties, and also on the chicken. Small, the house of in farms they used to grow layers. No, they feed on their blood. And that animal is not present here. This is the false vampire bat. We call it as Megaderma lyra. It feeds on frogs and small snakes, even the small spiders, centipedes like that and all. This Megaderma lyra is present. There is another one on the below to this Megaderma lyra. There is two species. There we call it as a Tophosaurus melanophagus, and we call it as a beard bat. The male have beard and the female won't have it. There is different kinds of species, and this one, that last one is Pipistrellus mimus, one of the smallest bat in the world. It will be grown for around 6 grams, and we can see it everywhere. In the behind our that is the electrical meter box. No, there will be a board behind itself, or also in everywhere in the colleges. These species notice board, behind notice boards. Whenever you go, don't disturb this animal. Just look behind the notice boards. These bats will be there. They have been present everywhere. We don't know where it is because it won't come out during the night daytimes. They are nocturnal in animals. After sunset only they will come out. They have a structure like that, and we can discuss about in the slides that has to be come in the next thing. So this will be present everywhere. If you are having a cow shed, there will be behind the tiles. It will be present everywhere. Small crevices in temples, they will be there. Church stones, they will be there. So these animals has been present everywhere. These are all some of the birds that has been present in the other areas of our environment, because there is another one, there is a Cariola picta, that is a butterfly bat. They call it as, it is a painted bat also. It will be called as, that is present in the plantain forest of the Western Ghats. At the base of the Western Ghats, these species are present. We call it as the Cariola picta. And other species, of course, has been there, and it has these animals are very small animals, around 13 to 14 grams animals. So they have been present everywhere in the environment. No, not a huge animal. Of course, that Megaderma lyra, the last one, no, I have shown, that will be around 30 to 35 grams. Other bats are very small bats that will be around 13 to 15 grams. And they feed on anything, that is fruits, insects, spiders, fishes, everything. They will be depending on. That only makes it to spread throughout the world. They won't have any specialized speeders. They are generalized speeders. They feed on anything what they get, and they live forever. But each species have its own feeding things. So if a species feed on nectar, it will surely feed on nectar. And another one, if it feeds on plant, another fruits, they will feed on fruits. So they have been present everywhere. So where they will be? There will be behind the Palmyra palms. This is the tent roost, we call it as, and the bridges, under bridges. Of course, all bridges, under bridges, that what we travel, everywhere they will be present. Small caves, mountains, crevices, everywhere. Tombs of temples and churches, everywhere. They use open trees also, they will be present. And also they live under the Palmyra palms, the curtain creepers, that what we have grown. The different types of palms, Palmyra palm, there has been processed fablivera, anything, any kind of palms, they will be living at a larger rate. So they can make caves on those animals, and they will be living. Second. Yeah. And this is what I have shown till now is the frugivorous bats. And of course, in India, there are 3 kinds of what I have shown till now, they depend on purely on insects. Once again, so these animals, there is a large difference between these insectivorous and frugivorous animals. What it is means, these frugivorous animals won't echolocate. When we talk about bats, we come into the word come into our mind is echolocation. They produce echolocation sound behind the hearing of the human hearing. They start to produce sounds and they navigate with that. But actually, the navigation, they have eyes. If you look after these photos also, you can see the bats having eyes. They have been eyes. These animals, if you take down this one, these fruit bats, they won't usually echolocate. They forage with the smell and also the foraging perches. They know it very well and they know their own area and they will forage to by the smell of the fruits and by the odor of the flowers, they can go and get their food. They won't echolocate. Actually, one species of the genus Rositus, that is below line, no, there is on blue screen, and that species, the only species that will echolocate in the frugivorous bat category. All the other frugivorous bats won't echolocate. They can travel with the sight itself. At night time also, they can use this sight and they will be getting on fruits. Only the insectivorous bat echolocate. And this another one. These are the largest species of animal. It is called as the Thiruchirapalli giganteus. I did my PhD on this animal. This is one photography. There is a endochorial placenta. That is, the placenta is actually like that of a human being. More researchers has been, if you go to this MG University Marathwada, they are doing for the past in the 1940s and up to 2000, they have done on the reproductive behavior of these bats to a greater extent. How the development takes place, how they do all these kinds of reproductive strategies. And this photograph, we have taken it out. It is a complete photograph. The placenta is attached with the pup. We call the young ones as the pup. Here is one. See, you can see the pup is hanging on a bat. And this is the mist net. So we captured with the bats with the nets, and the nets are called as the mist nets. And this is a Thiruchirapalli giganteus, which will be grow for around 750 to 1 kilogram animals. We will be aware of this animal because this is the animal that is behind Nipah. That is the virus that has come to India in the 2018. So these the thing behind this Nipah is this frugivorous bats. And how that it has been spread. I use the word bolos, no, because in India, no viruses has been present in these animals, because they have to share their virus with the other bats, then only they can get it, they can spread the virus. So India, there is no bats has been reported to have Nipah virus till now. But how it has come to that virus has come to the human being, no studies has been yet to be done, but the researchers are going on. But no clear evidences have shown that the bats spread these Nipah virus. But how in Kerala it has been spread in 2018 is still a question, because we used to say that taxonomy studies are very much essential, because nowadays the students doesn't like to do on taxonomy because nobody knows about the taxonomy. Whether it's plants or animals, the taxonomy studies has to be done. It has to be done in a wider manner, because I share you an incident in the Nipah. I have been working on bats. I have been called by the TNA, Tamil Nadu Veterinary, TANVAS, that is the Tamil Nadu Veterinary College and Animal Husbandry. They called me, university, they called me, and we shared some of the ideas. What it actually did means, in Kerala in 2018, the virus has been spread at Wayanad, the place, not the extreme part of the Kerala, there is a border there it has been spread. Actually, what it happens means, at first, at that time, it is the RT-PCR or the laboratories that now there has been everywhere in every part and every city we are having an RT-PCR lab after this pandemic. But actually, at that time, there is no such laboratories. In Tamil Nadu also like that, the situation. In every part of India, there is the situation existing at that time. What they have to do means, they have to get the sample and send it to the Institute of Virology, Pune, or to the Manipal University in Andhra. Of course, they have to send the sample to that place. There only they are having a laboratory of BSL-3 facility to done these kinds of research. Because see, every nation has its own laboratory conditions. In India, there is no BSL-4 laboratories. We can't do any research on the virus to a greater extent. So that we should keep in your mind when you are looking for these, where this virus can be cured or a vaccine can be find out. All these behind, all questions has to be answered with by keeping that in mind. What they have to do means each sample, because 18 people has been died at that time, 18 or 19 of course. What they have been to one person come, they say it is Nipah. The sample has to be sent to the Institute of Virology and also this Manipal University, and they shared it is a Nipah virus. So they come out and they say it is a bat. This is the bat that has been spread the virus by taking a bat from a well, and they have closed the complete well. Actually, what it is means, the virus can only spread by a frugivorous bats. They take down the sample of an insectivorous bat. Animal the open trees. So the taxonomy identification has come to our mind only when we are doing such kind of researches only. We can have the ideas, sumptuous amount of ideas. If you take down, for example, if you're doing research on sponges in the marine ocean, see, there is no one to identify the sponges, because sponges we are using it as a medicine, anti-cancer properties, and everywhere the research has been going on. But no one knows to, because very minimal people only know about the taxonomy and what is the species. They just go to the marine, they dive into the ocean, take all the sponges and grind it and do all these kinds of chromatography work and come out with the sample. But behind that, we should know what is from what species we have taken down these kinds of research. So taxonomy is that much essential. But everywhere we go, we are neglected for doing research on taxonomy. Why you are doing research on taxonomy? You can go with certain kinds of research, and that the funding also we denied by doing these kinds of researches. That's the thing, because these has to be answered at first. Then only we have to go for the next thing. So when you have an opportunity to do on taxonomy, don't hesitate. Do it. It will give you a great future. But this is an animal that what we have been talking about. And these animals, that is the Theropus giganteus, it is having a bigger eyes. This species won't ecolocate actually. There you can find, it will be for 24 hours, it will be in the outer environment. It won't behind a shelter. It will be present in the open trees for 24 hours, whether it is rain or any kind of season, autumn season or summer season. In Thirunelveli, it will be around 41 to 42 degrees at the deep summer, but it will be hanging in the trees at that time. We even can't stand in the heat, but that animals can live at that environment. So that animals are having such a thing. How we are doing studies by population surveys, counts, and that video shows how we capture bats in the the palm trees. There is a Cyanopterus sphinx, which is actually one of my scholars is capturing that species from, that is a hoop net. We use different kinds of nets for different species, because by using these surveys, we will be having anabat bat detectors that can detect echolocation sound of bats. When you fix the anabat bat detector in a echolocation detector in a field, you can actually understand what are all the species that has been coming through and how many ultrasonic sounds has been recorded. And we do such research, population surveys. We should understand where these bats are. If we have a complete data on the southern part of Tamil Nadu where these bats are present. Actually, during this also they have been called by the Tamil Nadu Veterinary University for this species where they have been, and they get the data from us. So the population surveys are there, where how we have to capture by using mist nets, harp nets, and trip lines. We use these category of products for capturing bats. And then this echolocation. So there is a small animal, there is one of the small animal in the world, which is Pipistrellus dormiti. It is around 6 grams. You can see these animals everywhere. This is a very small animal, the echolocation. When echolocation come into mind, actually these microchiropterans only echolocate. These small animals can echolocate. It can be echolocated at a frequency of around 35,000 to 65,000 cycles per minute. That we call it as the, so the sounds will be like that. That is kilohertz. That's around 30 to 60,000 kilohertz. It can produce a sound, but we can hear up to only 20 kilohertz. So these animals are very much can do echolocate at a very faster rate. The echolocation sound by means of giving sound or by tongue clicking. where when you click your tongue with the phalanges, the sound will be produced. No, like that only these animals are producing sound. Some of they will be doing that, and some of they will calls as normal or how we are produced. They have a good echolocation sound. So not all bats echolocate. That we have to keep in mind. That is the bats that depend on plants won't echolocate. The only exception is Rositus. That is the animal that I have shown you in the previous 3 or 4 slides. And by echolocation, giving echolocation sound, what it gets? The distance of the moth or the insect it is going to eat, the angle, in what angle it has been flying, what is the absolute size of the animal, what is the position it has been flying, how much elevation it has been flying, and the velocity. So with a small single call, the bat gets all these things. So I have shown a graphical representation of how it captures a moth. It produces a sound. And one second. Hello, can you hear me? Excuse me. Anyone? Please respond.
Yes, sir. Yes, yes.
Okay. I thought you was calling me. Sorry, sir. Because my screen is, the entire screen has been filled with this. I can't see you. Okay. Let me continue. Okay. These, if you take down, the echolocation has been produced. And there you can find one moth flying. No, there is a small butterfly has been flying. It won't directly capture the moth. It produces the sound. From it, it gets all the information. Distance, in what angle it is spreading, the size of the animal, and what is the position, and also the elevation and the velocity. So by producing a sound, it makes it detects its prey, and at flight itself, with the it captures with the patagium, that is the skin that is lying between the legs, hind legs. It catches and feed when it is foraging itself. So such things will be takes place, and so one animal, that is the one part. One animal, if it has to be completely success in the environment, it has to utilize the environment to its extreme level. How it has to be adapt to the environment. That is the first part. Then how it can be flourished throughout the environment, reproduction. That two things are important for an animal. One is the foraging, how it uses the foraging or opportunity. Then how it spreads its genetic to the total environment. So how it's spreading means we have given out the mammalian social organization. Depending on the reproduction, it can be classified into 4 types. It won't be there for the foraging, solitarily foraging primates, but we can look it for this one. That is the monogamy. That is the one male and one female. The animals will be like that for the entire season. Polygamy. Males mate will be with same female group for the entire season. We call it as polygamy. And there is one is polyandry. That is the females mate with same male group. And promiscuity, multi-male and multi-female. So this organization will be there, and it will be present with all the animals. But dogs, it will be there. By cats, it will be there. Each and every animal has its own depending of these things. How they are utilizing is still a question. But what I am coming to say is all these social organizations are present in bats. One species will go for monogamy, another species will go for polygamy, another species entry, and another species will be going for promiscuity. They are reproductively very successful animals. That's only it has been spread throughout the world at a greater rate and very successful. If you take down, this is a study what they have been done on Thiruchirapalli giganteus.
Sorry, excuse me, sir. Slide is not visible.
Now also?
Yes, not visible, sir.
Whether it is visible only, they have to pin the speakers.
Where it is, that one?
Now visible, sir. Visible only. Just to pin
Okay,
this.
right.
Yeah, it's visible only.
Okay.
Thank you. Okay.
Thank you, sir.
Shall I continue?
Yes, sir.
Okay. If you take down this one, let me take it. Okay, this is the Thiruchirapalli giganteus, one of the bat. If you look after the tree closely, you can identify the trees are same. Both trees are same. That has been taken during 2 different seasons. One is a breeding season, another one is the non-breeding season. The above one is a breeding season. The below one is a non-breeding season. If you take down a map, it shows the 2 districts of Tamil Nadu, the Thirunelveli and Tuticorin. That is the penultimate state of the Tamil Nadu state, at the last borders that has been there in the Indian world map. If you take down, there is a blue line, that is the river. At the base of the river, these animals are there. That is the Pteropus giganteus. Keep that in your mind. Always you look after these animals throughout the India, and they will be hanging in the open trees. If you look after whether the trees, it will be nearby an protective environment. A nearby police station will be there, or nearby a guarding forest environment will be there, and it selects it residential place for around 100 to 200 years by residing in this. It is having a high roost fidelity. That it used to roost, but India in a protective thing only. So it will be very hard for us to hunt this animal. Actually, hunting it, it chooses its roost like the places it is completely protected. Actually, why I have given this one is, if you closely look at the map, there will be 4 red dots, and that places we call it as the breeding roost. And the blue dots, that we will be calling as the non-breeding roost. Actually, what it happens means, the non-breeding roost, the females will be there, and the red one, the male will be there. At the mating season, it will be taking around the September, month of the September. From the males will come towards the females. Sorry, the females will flow towards the males. In usual phenomenon, what it actually happens means, males will chase down the females. The successful mating pattern will be like that. But here what it happens means, these females will come towards the males. Here the cycle, we call it as the estrus cycle, because the reproductive pattern, it can be classified into 2 types. One is the menstrual, and the another one is the estrus cycle. If estrus cycle means when the bleeding occurs during the phase, we call it as an estrus cycle in the females. When the bleeding takes place, it is a menstrual cycle. When the bleeding doesn't take place, it is an estrus cycle. It is an animal of estrus cycle. What it actually takes place means, the females will move towards the males. When the females comes toward near the males only, the males' reproductive activity will be triggered and the testes will be prominent. So then only they will be reproductively active. We studied such kinds of researches, and we get on project. Next one, I will come to that one. That is one important thing of this animal. So when you look after anywhere of these Pteropus giganteus, keep and look after it. At the breeding time, in a place in Thirunelveli, that is where I have did my research, the population of the bats will be around 20,000 in trees. There is around 16 trees in the one place. The trees are completely occupied bats, and it will be around 16,000 bats. The total bats will be moved towards the trees. So such a thing is there. So this is another fascinating animal. We get on project on this animal. If you take down, there is one small gland above its leaf. There is begin nose leaves. There will be nose leaves, and then one small gland will be there. We call it as the nasal gland. That one. In the gland, what it will be, the secretion. The gland will be there only in males. The females won't have these glands. If you take down the down pictures, they are glands, frontal glands. Females absence. Subadult male, it will be start to develop. And all these cases will be like that. What it will be happen means, these animals is a gregarious species that will be live around 150 to 200 species in a simple roost. They reside only inside a home or a temple roost. They will be depending on caves or some other places, and they will be roosting there. These males will produce these secretions, and what they will do with these secretions means, they will mark their females, their places, and they got their females. So with this marking efficiency, they will mark their females. Females won't have it. And another thing has to be come into mind. See, this is an animal that have its female pups. If you take down the last picture, a pup will be attached to an adult bat. The pup will be hanging in an up-down position only. Always bat hangs in up-down position. Of course, the pups will be hanging in its own mother in an up-down position only. There will be a small teeth-like structure. It will be bite on it. Actually, they used to weigh. It's a monkey. If it catches, it won't leave. Like that, bats, if it bites, it won't leave. See, Tamil, there is one slang in Tamil. I use just one thing. They used to say, "Korangu pudi oval kadi." The bite of a bat, and also you can't escape from a bite of a bat, and also the catch of a monkey. It will be like that. They will catch us like that, and it will be bite its mother like that. When it is taking for the foraging activity also, these animals will take its own females, their own pups with them. and they will bite in the teeth areas and will be hanging in an upside direction. The young ones won't leave with mother. By the bite only, it will be completely present in the females. At certain periods, what it actually here happens means the weight of the pup increases. We call it as neonates, and the neonates, they are at that stage, the weaning stage comes. The mother bats will start to leave its young ones in the roost and moving to the foraging activity. When they come at night times to feed it young ones, from the 150, there will be hanging small bats of around 100 to 150. They can find their own pup by means of sound and smell. That is an important thing. No. So they can also say each one will be similar. We are having a different thing, and our mothers can take the bats. All the animals will be looking similar only. You have to use tagging. We use the different kinds of tags to tag these animals and study. But their mothers know their young ones and they catch them exactly. Because there won't be any differences in them. So such a thing is there, and it is another fascinating creature. So they are such a way these animals has been spread throughout the environment, and they are reproductively success, and also they are foragingly, and also the using the environment to the maximum. They are success at all levels. So with that two part, I come to the next one, that is the viruses, bats and viruses. Due to this pandemic, we have to share some of our ideas. When we get an idea about these animals, we can know how hard it is to find the cure for these diseases. See, actually, bats and viruses, there is around 7 groups of viruses are there. There is DNA double-stranded viruses, single-stranded viruses, double-stranded RNA viruses, positive sense single-strand RNA virus. Actually, these only the coronavirus comes. The positive single-stranded RNA viruses, negative sense single-stranded RNA viruses. There is the Nipah and Hendra and Ebola. The Ebola there comes in the African countries, and the Nipah is also in African countries. And actually, in the past 5 years, the WHO have been giving caution throughout the world. The diseases are going to open up. The main one, Ebola comes, then the Nipah comes, then now this one, that Corona comes. The information has been already given to the countries by the WHO. When Nipah, before Nipah comes itself, WHO give a caution to the Indian subcontinent that there is going to be an outbreak of viruses in the country in the next 2 or 3 years. So they are doing research on, we are not doing research actually. A few people around the world are doing research on us, but because we don't have laboratories, you should keep that in your mind, because there is no approved BSL 3 or BSL 4 labs. Only we can do research on this, what we can say, that one. We can do on that. At the Pune Institute, we can do research on the viruses. That is the necrosis virus. We are doing some research. So the other than that we can't do, because if you take down this one, Hendra viruses is there. The only approved lab to do research on Hendra, it is from bat to horse to human beings. That is the Hendra virus. So bat, they will come to horses and then to the human beings. This research can be done only in Germany. They are having that BSL-4 laboratory. So no other laboratory these researches can be done, because we can't grow a virus in a laboratory. That it needs very sophisticated. We have to change according to the change in the norms and also the equipments, because we can't invest on RT-PCR for throughout a time. It costs around crores of rupees. Now what we are getting is around 500 samples can be stored, and one instrument costs around crores. So like that, there is on MERS viruses. There is the Middle East Respiratory Syndrome, and also the another one, that is the plague. All these comes under the that is the group 4-1. That is the group 7, Hepatna virus. These viruses, as now when you look at when you are continuously watching on coronavirus, another viruses has been outbreak in the China in the last month. That is the virus, Hepatna virus. So why I am giving introduction means all these viruses are present in bats. You should keep that in your mind. Actually, what they are saying is the mumps and measles we are getting before thousands of years, that virus is also coming from came to the human beings through bats only. Bats have been only acting as the transferring agent of these viruses to the human beings. What So when you come to this zoonosis, two animals will come into our mind. I have compared with two only, the rat and the bat. If you take down the first animal, that is the highest number of zoonotic viruses, where it is means rat only. Small rats, it will be there. Calculatedly 179 of which 68 can attack human beings. There is around reported of 179 viruses, of which 68 can attack human beings. And one such example is leptospirosis. When you take down bat, that is the next animal to the highest number of zoonotic virus to rats. Calculatedly 137 species are there. 61 make disease in human beings. Now the research has been going on. But the strains there, it is around 300 or 400 strains. But the viruses, we have given name for around 137, of which 67 can make diseases in use. The examples are rabies, Marburg virus, Hendra, SARS, Nipah, and these kinds of viruses. Why these animals? Why I have compared these two? For example, when an animal is very active, we have done small experiments on the depending on temperature and all such kinds of things. What is the Q10 value? How it has been small experiments in BSc classes. No, that's when an animal is very active and have fast metabolic uses. This is the simple phenomenon. When an animal is very active and fast metabolic rate, its lifetime reduces. But if you came to that animal, rats, which are similar to that of bats, which is a small mammal, both are small mammal. Rats lives only for 2 years. If you take down bats, they can live up to 40 to 60 years. How it is? It is against nature. No, bats are very fast and very aggressive animals, but they are living for around 40 years. Whereas the same animal, that is rats, which live for only 2 years, of that category, how it actually comes to your mind? There plays the major role by the viruses. Why they are incorporating such amount of zoonotic viruses is, this is the thing. The first one is the temperature. The body temperature goes all the way to 42 degrees. When it is on flight, at flight, the temperature of the bats goes to around 42 degrees. Even though the bat had given space for those number of viruses, how it check the viral load inside its body means, the first thing is the temperature goes to 42 degrees. At that time, these viruses find it difficult to live and the viral load will be decreased. That's what we are very much bothered about this animal. Actually, these viruses lives in 42 degrees Celsius. When it comes to us, our body produces the temperature of around 40 degrees. Actually, the virus lives at 42 degrees. So it is nothing for it. And that's what only our immune system finds it difficult at the initial stage to fight this virus. And then another one. Why that amount of, how that amount of temperature is produced? The heartbeat of a bat goes to around 1000 beats per minute. Imagine it. We are having 80 to 90. That is depending on exercises or walking, what we make, the beats increases. But for these animals, it will be around 800 to 1000 beats per minute when it is flying. What will happen means the metabolism will be very faster. They will feed at anything. They will forage at that. They will feed on tons and tons of insects in an agriculture field when they have been flying, because they need energy. And when energy is produced, the waste will be accumulated at the large rate. We call it as the free radicals. Actually, it also happens when we go on aging. The free radicals accumulation will be very more, and our cells will got ruptured by means of these free radicals. Actually, oxygen free radicals will be there. When you make these free radicals to decreased, we have to depend on antioxidants for our living. Otherwise, we start wrinklings at a very faster rate. At the age, when we get the age, the wrinklings will be, skin will be wrinkled. But what it actually happens with this bat means, how it overcomes it, this one, because when it comes of these free radicals, let me give a small data to these free radicals. Actually, the free radicals have been reacted with the organs inside our body, each and every cell, and our cell will get ruptured, because our cell is made up of the saturated and polyunsaturated fatty acids. Cell has been covered by these saturated fatty acids. When we speak on it one, they actually, these we call it as the oxygen reactive species, because we used to say our cells are covered, cell organelles are covered by these fatty acids. And actually, what it happens means for us, the cells are covered by saturated and unsaturated fatty acids. And that to have another thing keep it in your mind, the saturated are single bonded and the unsaturated are double bonded at a larger rate. That is the carbon-carbon bonding. There will be double stranded bonding. And when it is double stranded, double bonding, it will be easy for them to break down. So our cells easily break down these free radicals and we got aging. But what it actually happened with the bats means they are the unsaturated also it is single bonded. The maximum is single bonded. Even though they also have double bonded, the maximum is single bonded. So it guards its organelles. And another one thing happens there, when the free radicals accumulation takes place, there is a mob waste in cells faster. How we clean our houses with a mob, the bat actually mob out all the free radicals outside its cells. At that time, it expels out the viruses too. So it keeps the viral load in check. That is the first in temperature, and also by cleaning of the free radicals. Along with the free radicals, it expels out the viruses. And then another one is the sting interferon pathway. It is another pathway. What is it? The interferons, we know they are signaling, cell signaling materials. So when a virus is enter into a cell, the interferon, it is having a different mechanism. All the information has been spread to the nearby cell. We call it as a shift attack. And what it gives is all the cell will get activated and a new foreign particles has come into our cell. We have to guard it and the cells will become very stronger. Cell wall will be very stronger. And what it happens means these viruses find it difficult for the replication. And it also shows no immune activity against these viruses. In such a phenomenon, these bats uses the viruses for itself and makes it cells and immune activity at a stronger rate and living for a longer age. So its longer living lifestyle depends on the viruses what it has. So they are having a mutual relationship. See, if you're speaking on sting interferon pathway, see the similar thing is happening inside our immune system. Actually, we are having certain people has been infected by the coronavirus and they won't show any signals and any infections and they can recover it. Some of them are having an activity of these chemical signals at a very worst rate and the body finds it difficult to withstand this immune system because like that sting interferon pathway also, a similar one takes place inside our body and our cell walls are blocked. So that is also chemical signals. The immune system is entirely different with how it acts in a bat and how it is acting in a human being. So we are getting a more number of diseases because usually they will say they take one person with a corona in the next 24 hours he dies. And this signaling makes all the changes of these viruses. So can bats infect human detectively? Because if we keep kept in your mind, no such data or no such research or no such disease spreading has been studied in India, or we haven't come across such things in the past 100 years. Of course, we can say 100 to 200 years. We haven't come such a studies because it won't take place because there is lots of kinds behind this one. Because bat won't directly infects human host. Until now, it has been there. But the new researchers, because the research has now changed all its actions. Because one country want to prove that we haven't done anything to their human beings. We have done any research on bats. We haven't spread the viruses. Another country want to prove, no, you have been made, it is a synthetic virus, you have made it. So such things have been classified between the different countries. But actually the research till now, what it says means that it should have an intermediate host. That is, bat has to spread its viruses to the intermediate host, then only it can spread to the human beings. So there has been different kinds of viruses. There is SARS virus, there is MERS virus, there is SARS-CoV, all these kinds of viruses, they have to be spread to the human beings only through an intermediate host. The bat can't directly spread to the human beings. How it's spreading means, I used the bolos at the second slide, no. See, what bats will do means, when they are immunologically or what they used to say means when bats are disturbed at a larger rate, the habitat destruction takes place, and they have been disturbed at a larger rate, their immune system fails. At that time, the viral load will get erupted. So what that means, its saliva or the fluids will get these viruses. At that time, when it feed on any of the fruits and it puts its spillovers, we call it as bolos, no? It brings the juices and put on the bolos. At that time, when it is fed by an intermediate host, if you take down this one, there is a camelids, there is cow, there is camels, MERS CoV. It comes in the Arab countries, Middle East countries. It is called as the Middle East Respiratory Syndrome. Yes, similar. All the infections are similar to the coronavirus, and SARS is also like that. It also comes in the early 2000s. So there is a camelids, there is an animal, there is one civet cat, that is the intermediate host for SARS-CoV. I discussed this one here. Because at that time, what it happens means when SARS comes out, everyone like that, like this only. The world has been doing research, and they find a vaccine for this SARS-CoV. At that time, what it happens means a lesser virulent host, they have find one, HCoV NL63. It is a lesser virulent one, similar to SARS strain. And what it actually happens means, if a person is infected by this HCoV NL63, he won't be infected by the SARS-CoV virus. That acts as a vaccine. So that doesn't infect like that of a SARS-CoV. With mild infection, the human can be lived. But when he is infected by the SARS, he will be infected at a larger rate, and his life will be in a massacre. So at that time, they used this HCoV NL63 as a vaccine to this animal. Now also they are finding whether there is a less virulent strain of this coronavirus. All these are coronaviruses. That what we have shown is there is HCoV, OC43, all these are coronaviruses. Now that is the swine flu, SARS-CoV. That another kind of viruses that it comes through the pigs, camelids, and also these civet cats. So the bat won't directly, it won't spread any disease to the human beings. But in the past 1 year, 1 month, we have been seeing some papers. They are representing that bats can directly spread diseases to human beings. But till now, that research is under a posting only. But of course, we need this intermediate host. Why an intermediate host has to be there? There comes another example. With this slide, I will conclude my sessions. So this SARS-CoV, that is the SARS virus, that is in the 2000 outbreaks. There is one bat, we call it as Rhinolophus bedomi. These two species are, of course, present in India. I am going to speak about these bats. These are the bats that is behind this coronavirus and also the SARS-CoV. That is the SARS-1 and SARS-2. Our NCOV-19, we call it as SARS-2. See, SARS-CoV, what is this Rhinolophus bedomi? The bat cell, it lives in the hill regions. Because you should keep that one thing in mind. These are very small animals, around 13 to 14 gram in size. We won't eat it. Anybody won't eat it. Because it is very small animal, though, where you have to shred flesh from its skin, like a small animal. So the Rhinolophus bedomi, it is a small animal. It is eaten by this palm civet. The intermediate host, no. The intermediate host, the viruses has to give it to the intermediate host, and then it can spread to the human beings. If a human feed on this civet cat, he can be infected with the virus. Why it needs an intermediate host? If you take down a complete strand of a NCOV-19, it consists of around 30,000 base pairs. So of that 20,000 base pairs, we call it as the virus, actual virus, that is the NCOV-19 virus. Another 10,000 base pairs, we call it as the spike RNA. So there is a spike RNA. Spike has to be there. And the spike only can transfer or infect the ACE region of the that is the region that it can attach with the human cell. So the spike region, these bats viruses, they have studied different kinds of virus. Actually, what they are saying is these virus that present in the bats doesn't have the spike RNA to come to a human being. I had thinking that I can explain to you correctly. That is, the Rhinolophus bedomi bat has got the virus of SARS-CoV. It can't be directly transferred to a human being, because the virus don't have the spike RNA that can bind with the ACE region of a human cell. it can only bind with the ACE region of an intermediate host. So at the intermediate host, after getting a lot of mutations only, then it goes to a human host. So such a thing has been happened with this SARS. On in mind, we haven't find this vaccine for SARS-CoV. If you take down this for the past 100 years, in the 1920 we have got the that one, Spanish flu. Then after 20 to 30 years at the Second World War, the First World War, we have got the Spanish flu. At the second, we go for the Asian flu. Then another one, we go for the SARS, then MERS, and then NCOV-19. No vaccine has been found till now for any of the viruses. So we are in that thing. See, at that only Spanish flu also, it is there for around 2 years. Now they are also saying this NCOV-19 will be there for 2 years in our environment, and it is going to be there. We have to adjust ourselves to live with these viruses. That is the only thing what we can do now as far as now, because we can't hold on our life for a longer time like this. No, then another one comes. That is the Rhinolophus affinis. It is a small animal. It will be living only in the hilly regions. Of course, in the Western Ghats, this animal is here. At 1999, we have captured this one bat. That is the Rhinolophus affinis. And its intermediate host is the pangolins. They say there is 2 hosts actually, pine cone soldier fish, that is one fish variety, and after that they say it is not one, that is the pangolins. Actually, pangolins from the Indonesia, they are saying that carries these viruses. Actually, what it happens means in Chinese cuisine, they eat on these pangolins. So the pangolins has been infected by this virus and they have been spread to the human beings, but no one speaks on it, how it has been transferred from the bat to the human beings. And no studies say that which species having such a viruses to infect directly a human beings, because it need an intermediate host. In India, we won't eat palm civet or pangolins. We won't feed on pangolins. It is not in our daily or regular diet. So we don't need to worry. In the past 200 or 300 years, we haven't been infected by any of the viruses directly from the bat. What are the challenges to overcome? There is first thing is Bats Zoonotic directly doesn't infect human cells. So we have to find which one is the intermediate host. What does that spike RNA actually doing? Then actually virus spots from bats and spike region from intermediate host. That is a modified. How many mutation does takes place in the spikes? Actually, we are doing vaccines to stop down the spike region action against these ACE proteins. Why we are saying we have to take care of the elderly means when we start taking that tablets for blood pressure or increase in blood pressure, actually these ACE regions will be activated at a larger rate. When the ACE region are activated by the tablet, it will be easy for the viruses to hang with the ACE region of the human cells. So they are very prone to these viruses. That is the aged people. So those who are taking diabetic or also diabetic, of course, diabetic and that BP tablet for a longer time are very prone to be these kinds of diseases. Then viral parts. If you take down this one, 2/3 of the virus base pairs. If we say 20,000 base pairs and 10,000 base pairs, 20,000 is the virus and the 10,000 is the spike RNA. If you take down that virus, if it is cleaved at the spike RNA, that virus part alone can live individually. And that's what we are getting results at certain times. That virus part has been found in the drainages and ditches, the ditch water and the drainage water of the hospital has got these viruses. We know the virus can't live individually without any host cell. How it is living? Actually, we are using the virus RNAs. The RNAs are actually have been cleaved in the laboratories, because in the laboratories when we are running RT-PCR and all those things, it is cleaved. It has to be cleaved. The 30,000 bases has to be cleaved, and when it is not properly properly we have been destroyed it, actually that will come into the drainage system. Actually, of course, in the last 2-3 months that Chennai hospital has got the viruses in the drainages. Of course not. That are the remnants, actually the cleaved remnants. They are also active. Actually, what are they saying is if it is inside a cell and a virus is cleaved into 2 halves, the spike and the RNA, and the spike and the viral RNA can live for a longer time. So these animals shows a different activity in and against an immune system, because in cell lines it shows a different thing. Actually, we are doing research with these cell lines, animal cell lines. We are growing in the laboratories, and we are showing growing the culture and studying. But actually, actual working condition differs with the immune system. When the immune system is aggressive, these viruses are more aggressive. When the immune system is less aggressive, the viruses doesn't spreads at a very long, because our immune system is entirely different. It is very aggressive against any of the foreign particles. If you take down bats, they won't have such aggressive immune system against these bats. Bats are, of course, infected by this virus. That is the Lassie virus, one of the virus. It will be infected the bat at a large numbers. Most of the viruses won't infect this animal. No studies in the world that speak on activity of a virus in bats. Actually, what activities viruses are doing in bats, and also the next one we have to come now, intermediate host, how it is, and the host cell. Actually, this is done by the European laboratory. They gives out until or unless we found the animal how it works in bat, and then viruses in intermediate host and host. Until then, it will be hard for us to find the vaccine for these animals, and then only we can challenges and we can overcome this. Until or unless we can use in the laboratory by drawing on the computer, we can look after these are all the RNAs, these are all the spikes, and we can put down any restriction sites, and we can cut down, and then only we can. It is easier for us to study in a computer. When it is in a wet lab, there is entirely different. The animal shows, the virus shows entirely different characters, but everyone is doing research on this animal. Yesterday also one of the vaccine from the China comes out. Before that also one from the Russia. But does it goes to changes anything in the next 6 months or so? It is very hard to answer, because all the experts, landed experts, are saying that it will be very hard for us to come out of these viruses. So for the past and next 2 years in the developing countries, and for the next 1 year for the developed countries, the virus will be there. But how they are mutualistically behavior? Because you have to, I am looking after this in a bat's perspective. Actually, it is an animal. It wants to live. It is living by means of the virus. So for a human perspective, it is spreading diseases. We have to stop that one. So, but it on bats, viruses gets placed to replicate. Why? Because bats are always hide from the sunlight, direct sunlight, where viruses can spread at a very large rate. They give an environment, dark environment, viruses can grow at a lot. What about the bats? What all are the benefits that it gets because of it inhabits, give place for the viruses? There is no cancer in bats. There is a first thing. They have a live longer life. That is another thing. Everyone needs it. No, we too needs it. We doesn't need cancer. We have to live for a longer life, and we don't need an advanced immune system. So why not? Bats cannot be a reservoir for a stressful virus if it gets such benefits. Only thing what we can do is we have to get rid of these animals, thereby giving it space to live, and we live in another environment. That is it. They should not be disturbed. So we need a study on evolution. Actually, we forget this subject because nothing in biology makes sense except in the light of the evolution. Without knowing a cell, we are studying on the DNA. We should know about the cell, then only we can get a study on the DNA. We know about the development of a cell, then only we have to go for the DNA. At first unit itself in colleges, we are having a DNA. What it is going to do? They don't know where is a cell, what is the importance of a cell, what are all the cell organelles, but we are going for they know about the DNA splicing mechanism, RNA splicing, everything. But what we doesn't know is the basic of the unit of this biology, or the biology, that is we don't know about the genetics, we don't know about development biology, invertebrate, chordata, everything. We should know about that only, then only we can go for the cells and what is the importance of DNA and why it has been changing. So nature teaches you everything, as said by William Wordsworth. The name itself, Will I Am Wordsworth, that is the name there. I have been denoting it. The nature teaches you everything. With this, I conclude my session. Hope you all understand, and you can share some of the questions that you want, and I will be ready to answer. Thank you.
Thank you, sir, for your nice presentation. And our Bats Zoonotic with many examples. It was really very interesting, sir. Now the session is open for discussion. Any questions from participants? you can ask directly to the resource person, or you can put chat box. Sir, there are some questions in the chat box.
Yeah, I can.
Would you read, or I will read?
Oh, no, not necessary. Phylogenetically, to what levels are genomes of bats and COVID-19 are similar? Actually, it is around 92%. It has been proved. Result, one of the strain has got around 92%. Economic and medicinal values, of course, there is no medicinal values of these bats. Sure, I'm pretty sure about it. But economically, we are doing research on that one, because how they are economically important for this agriculture control pests. Because see, there are certain flowers that blooms at night. I give you a question. You can check after your internet. The flowers only bloom at night. For example, I give you one example. There is Cepa penandra. We call it as the silk cotton. So that a plant, no, that plant only blooms at night. Pollinated by bats. Bats are the only pollinating agents for these fruits. So these animals, where these plants will go. There are different kinds of plants I can example, but I give you one quote. You look after and see how many plants are there, and they are waiting for the bats to get pollinated. Because nocturnally, flowering plants can be only pollinated by bats. In certain islands, these animals, Australia and all, these animals are kept as the keystone species. They should not be disturbed at any time, at any level. Yeah, that's that much that they are very much economically importance. But medicinal values, of course, they are giving for most of the asthma and all they are giving, but it doesn't show any medicinal properties. Actually, we don't need to depend on its meat for as a medicine. Actually, all these problems comes with that only. They used the bat of medicine of various in Chinese traditional. They use animals as a source of medicine. Actually, that makes all these changes. Yeah. What is ultimately going to happen means all the research funding for these kinds of researches will be stopped at a very particular rate, because it is not necessary. That is the thing. What it is going to be happen. But if you get an opportunity to do research on basic sciences, actually what we have to do, and we are very familiar with these kinds of things, because who are all the great biotechnologists were once they have been doing these basic researches. So try to do it. I take this an opportunity. Yeah. Any other than that? 2 questions only there. Thank you. Hope you understand. You can ask me in any language.
Any other questions from the participants? Please.
Sir, this is Masilamani. Do you find any diseases in the nearby areas of where the bat population is more in the trees?
Actually, no such reports were there, sir. In our area, no such researchers were there. Actually, when the Nipah outbreaks takes place, we are some of the peoples from the National Institute of Virology. They put a lab at Kerala, because the Nipah comes in Kerala. When they studied about these viruses at that time, they show it doesn't show any of the viruses, Nipah virus, in its body. So no diseases has been spread by bats until now in India.
Thank you, sir.
Any questions from audience? Participants? I think it's no questions from participants, so I can come to the conclusion, the end of the session. Thank you, sir.
Yeah.
Now I.
So Ramesh is ready for Thanksgiving.
Sir, what is that?
Sir, Ramesh ready for Thanksgiving.
And great. So thank you participants for your active participation in this session.
Yeah.
So now,
Let me
now I
continue, sir. One minute. Yeah.
yeah.
So much of you are in this session. I am Sudhakaran once again. So much of you here, there is around 15. I put a question to you. See, since we are biologists, we know that the bat increases its temperatures to, I won't say the answer, let you Google it and find it out. Bat increases the temperature to 40 degrees. Actually, the virus is check in control. That is the thing out of what I said.
Yeah.
But if it is the bacteria is also dying, no, it should have its own flora. It will also die. Then how it gets the digestion and all those things will take place, metabolism takes place inside its body. You Google it and find the answer. I know the answer. If you have also known, this session is useful for me for sharing some of my ideas. Thank you.
Very good, interesting information, sir.
Okay.
We will search it. Now I call Dr. M. Ramesh, Professor of Zoology, to propose a word of time.
Yeah, good evening to all. It's my pleasure to thank you everyone at this junction of this national webinar on our bats or zoonotics. First of all, I thank our principal ma'am, Dr. J. Sugandhi, who encouraged us to do the webinars during this lockdown period, and also thank our IQAC of Maria Rivia College team for the smooth conduction of these webinars. Next, I thank our head of the department, Dr. S. Uma Mageshwari, convener of this webinar, for her wonderful welcome address during the beginning. My wholehearted thanks are due to the resource person of this webinar, Dr. M.R. Sudhakaran, Assistant Professor of Zoology, Sri Paramakalyani College, Alwarkurichi. He has elaborately explained the factors of bats, like its feeding, breeding habitats, pollination, especially its viral disease transmitting processes, how bats are acting as intermediate hosts, and so on. Once again, I thank Dr. Sudhakaran sir for his wonderful and informative lecture, sir. My special thanks to the organizing secretary, Dr. E. Moguddin Asghar Nawaz, and the co-organizers of this webinar during this pandemic period. Last but least, I thank the participants from various colleges, staff, research scholars, and students of various, not only zoology department, from various departments who actively participate in this webinar. Thank you. Thank you, one and all.
Thank you, sir. Thank you, sir, Dr. Ramesh. Once again, I thank all the participants and especially to the resource persons. Thank you, sir. Thank you very much, Dr. Sudhakaran, sir.
Thank you very much, sir. Dr. Sudhakaran, sir. Thank you.
Thank you, sir. Thank you.